touched on some of the work I did in my PhD, and this was around photodynamic therapy of various different gynecological neoplasias. And I was really fortunate that the bioadhesive patch that I developed was used in a clinical trial of over 100 patients with various different vulval and vaginal dysplasias and neoplasias. And we were able to cure 50% of the patients and achieve meaningful improvements in the conditions of the other 50%, such that their quality of life was greatly improved. And delivery of photosensitizers, I suppose, was what led me to the microneedles field, in that most photosensitizing compounds don't really penetrate biological barriers very well because they tend to be quite large molecules, and quite often they're hydrophobic in nature. So at the time that I was considering ways to enhance delivery, microneedles, through the work of Mark Prausnitz and some of the work that Sassano Pharma were doing, was really attracting interest. And so it was a natural step for us as a group to look at microneedle systems. And that's when I came up with the idea of making microneedles from hydrogel forming materials. So these are materials that are hard in the dry state, but they're chemically cross-linked, so that whenever they penetrate the stratum corneum, they will rapidly take up skin interstitial fluid and swell. And this means that you have a continuous unblockable conduit between a drug reservoir attached to the top of the microneedle base plate and the dermal microcirculation. So for the very first time, you're not limited by what you can load into or coat onto the microneedles. And also, as a formulator, you can control the rate of drug administration by modulating cross-link density. So that means that the rate of transdermal delivery is, for the very first time, controlled by you as a formulator. So this opens up a whole range of possibilities for increasing the range of types of drugs that can be delivered across the skin. And if you think about the conventional transdermal patch market being worth in and around about $30 billion, but it's only based on 20 drugs at the moment, if you can increase the range of types of drug you can deliver across the skin, because now you can deliver drugs in high doses across the skin, you can control the rate of administration, then potentially there's a lot of value for the pharmaceutical industry, and important for myself as a registered pharmacist, a lot of scope for improving therapeutic outcomes for patients. Hydrogel forming microneedles, of course, can also be used in diagnosis of disease or patient monitoring, because they can quickly pick up biomarkers and drug substances from skin interstitial fluid, which, as we know, is in balance with the blood plasma. So even though we work on a range of innovative dissolving and hollow microneedle systems and even coated microneedle systems, I think the platform that we've patented on hydrogel forming microneedles has the greatest diversity and has quite a lot of potential for impact. And we've licensed that technology to a number of different pharma companies in terms of drug delivery, but also in terms of diagnosis and patient monitoring. And it's an area that we continue to develop within my group here in Belfast. And I think that historically, it's possible that the pharmaceutical industry considered microneedles as simply a vaccine technology for developing countries. And they figured that they couldn't make any money out of that, and maybe they didn't invest as much as they could as a result of that. And so really the microneedle vaccines field was relying on big donors like Bill and Melinda Gates and the US government funders to advance the development in that area, and the Wellcome Trust from the UK also contributing strongly. But I think now pharma are realizing increasingly that microneedles have a big role to play in drug delivery in general, particularly in delivery of long-acting medicines or in delivering of higher doses across the skin, both of which are areas that my group are extensively involved in with big pharma companies. The second big aspect has always been manufacture and being able to scale up the manufacture of microneedles. So I work very closely with LTS Loman, the world's biggest transdermal patch company, and they now have Europe's first and only manufacturing license for GMP microneedles. And they're making microneedles and using them in a range of different clinical trials and working with a lot of big pharma and vaccines companies to produce microneedles on a scalable basis. And then the next element is the regulatory considerations around microneedles. And I know that this is an area where Zosano and others have made considerable progress. And I think the regulators are learning a lot more about what microneedles are and are able to provide advice to developers at an earlier stage. And a lot of this has been facilitated by PATH's Center of Excellence for Microarray Patches, which brings together microneedle developers with industry, with regulators, with not-for-profit organizations, all working together to advance things like critical quality attributes for microneedles, think about microneedle manufacture, think about quality control tests, and also to advance the regulatory position. So even though you might think that many of these groups could be potentially rivals, they are all coming together under the PATH umbrella to help advance the technology. And this is how we're really going to see this as a field moving forward. I would like to see a microneedle product over the next couple of years. And I think once we see the first product on the market, I think we're going to see an explosion of products. And this really will continue to drive innovation in the area, but also is going to be of great benefit to patients. The challenges are very similar to the innovations that we need to drive home. So manufacture is one, funding from big pharma is another one, and then regulatory approval obviously is another hurdle that needs to be overcome. I think there have been numerous clinical studies and very high quality academic publications in the area. I think there's a real need for microneedles now, especially in the wake of the COVID pandemic, because I think that while vaccines will continue to be given with a needle and syringe in developed countries, and we probably will all need COVID boosters and may need to be vaccinated on an annual basis, this is taking up a lot of time for healthcare workers. It's also taking up a lot of resource. And then there's the big backlog in diagnosis and treatment of other diseases that needs to be overcome right across the world. So what I think is that a lot of medicines that currently have to be given by injection on a routine basis, so long-acting contraceptives, long-acting treatments for schizophrenia, HIV, they could all be given by microneedles in the patient's own home, instead of having to come to a healthcare setting where they're going to cause a burden by taking up time. They're also potentially could contract COVID or bring COVID in to the healthcare setting. So I think microneedles have a big role to play in developed countries in administration of routine medicines that ordinarily have to be given by injection, and of course in developing countries in administration not only of those long-acting medicines or high-dose injectable medicines, but also in vaccination, where there are a lack of skilled healthcare workers and there's a big problem with inappropriate use and reuse and disposal of needle and syringe type vaccine or injection products. And also, as we know, a lot of the COVID vaccines in particular need refrigeration, but there's been a long-standing problem in terms of maintaining a cold chain for most vaccines. So I see there being a big role for microneedles in vaccination in developing countries in particular. And I know that the work that a number of people in the field have been doing on measles and rubella means that may well be the first microneedle vaccine that makes it to patients as an actual properly registered product. Having strong mentors and people to look up to has always been something that I felt was really important. And I was very fortunate in my time in the School of Pharmacy in Belfast to have two really good mentors. First, my PhD supervisor, Paul McCarran, who had really good scientific knowledge and was very innovative when it came to building equipment to do particular pieces of work. And then also David Wolfson, who was our director of research and then head of school here. And David had a long track record in innovating new delivery systems for the pharmaceutical industry. And he also had a very keen eye for what was a good research idea and how that should be best articulated. I'm very fortunate that I'm still in contact with Paul and David, even though they've both now left the School of Pharmacy. I also was very much inspired by Duncan Craig, who was at the time a hotshot young professor brought into the School of Pharmacy at Queen's to drive forward research, and how he was able to build a research group that had a very distinct identity. And that was something then that really showed me as a PhD student at the time what was possible in terms of how you could develop your career and also how you could build a research group to drive success. You know, looking further afield, I was very fortunate to have spent a summer working with Professor Johan Moen in Oslo. And he was a great character and a pioneer in the photodynamic therapy area. So it was great, a real honor for me to go and spend time and work in Johan's lab. And his group showed me great hospitality, and we were able to publish a number of papers together. And this is something that I've used as a marker for my own group. And we would regularly host visitors from all across the world. Really showed me the importance of diversity in a research group, not only in terms of scientific discipline, but also in terms of nationality and cultural background. And I'm very proud that today, out of 36 members of my group here, we have 15 different nationalities. And that's something that has really helped to drive innovation in our group, and also makes for really interesting discussions around tea time and lunch within the team here. So those guys have all been really important. And I should also mention Professor Sandy Florence, who again has been a real innovator in the pharmaceutical sciences, but also a gentleman and somebody who, despite all of his achievements, has remained very humble and always has a word for young researchers anytime they meet him. And I was very fortunate to spend time serving as external examiner at King's College in London at the same time that Sandy was there. And I really enjoyed conversations with him, particularly around pharmaceutical sciences and the future of the pharmacy profession, as we met around the table, had lunch together, and then used to travel back out to the airport together. So I was really delighted to have the opportunity to spend time with somebody of Sandy's stature as well.